we do, off this latest biennium, kind of have a mandate to set up a more active scheduled monitoring program. We've done some kind of pilot studies on how that's going to turn out, with hopefully implementing it in this next season. All of these samples are grab samples, and analysis is through ELISA. Next slide, please. When OHA makes an advisory decision, this is the OHA's rule. They either use it based off of the data we provide or another lab, or they can decide to post an advisory based off of photos that would be later supplemented with analysis. And these are the recreational limits in micrograms per liter of the various toxins that are tested for an advisory. This is the response timeline that Kurt mentioned earlier. These are when the advisories came out. It started with the Willamette Cove advisory, as Kurt mentioned. By August 9th, we had a Ross Island advisory. And then by the middle of that month, as the bloom was exiting the Ross Island, we had to combine the advisories into one continuous advisories. By the end of September, we first dropped, or OHA dropped, the Willamette Cove advisory, and then the Ross Island advisory was lifted. This is more of a location-based view of our sampling efforts, complete with the results. Again, it started in Willamette Cove and spread to Cathedral Park. That was the first advisory. The second advisory in Ross Island was based off of photographs until we were able to sample. We sampled the whole river on the 16th. Looking at this data, you can kind of notice that Ross Island on the 16th, there are very high microcystin concentrations, 549 micrograms per liter. As we drove downstream by Duckworth Dock, it was down to 0.18, which is below recreational levels. But by the time we got to Lundberg Beach and the Ash Grove cement docks, it was back up to 184 micrograms per liter. And then back to Willamette Cove and Cathedral Park, it was back down to 4.3. This kind of supports Kurt's data that shows that it came out in waves, and we're kind of seeing that on the river through our analysis as it kind of pulsed down the river. And I think this is you, Kurt.
So yeah, so the bloom, lasted for what, 3, 4, 5 weeks, and then we sort of had to fall back to the diatom. Diatoms and green algae seem to really kind of become dominant. Some of the cyanobacteria here, Microcystis, we had some Delicospermum for the taxonomists in the crowd, and then also some Aphanizomenon. So we had sort of that Annie, Fanny, and Mike group there. But by and large, it were these more beneficial, I think, types of phytoplankton. So there you go. Thanks for hanging in with us. Glad to try to answer any questions.
Thank you very much. Do we have questions here? Theo.
James did have a question about, were there any concerns upstream from Ross Island Lagoon near Selwood Bridge, noting that the reversing flows at times occur there. We did have concerns. We visually inspected all the way past Willamette Park up into below the Willamette Bridge. We didn't see any blooms there, so they were not tested.
You have a question from the room here?
Yeah, question for Kurt and Nathan. Kurt, what depth was in the water was your monitor?
Pretty much right at the surface, within the first few inches.
Okay. And what do you know about the depth of water that is picked up by that tidal inflow and outflow out of Ross Island Lagoon? Do you think it just picks up superficial water or a range of depths?
Well, that's a good question. Desiree Tellis has been looking at some of the hydrodynamics there. It seems that is sort of towards the outgoing part of the tide, the river stage is coming down, and down and down and down, if you can sort of picture that. And then at some level, and probably wind is also part of the dynamic here, but as the tide goes out, it leaves the lagoon as well. And it sure looked like the surface of the lagoon was preferentially pulled out. So I think maybe that's kind of built into your question: is there like multiple layers that are getting drawn downstream? And it seems to be sort of like the surface, as the water elevations drop downstream, it just sort of sheets out, I think.
Good, thanks, Kurt. Hey, Nathan, in those satellite pictures, you focused, of course, on Ross Island Lagoon, but there was a huge bright spot just to the southeast of the Holgate Channel. Any idea what that was?
Yes, Oaks Bottom. So the USGS React tool hasn't been calibrated for Oregon yet. It's developed out of Idaho. And there's quite a bit of azolla, duckweed, and some cyanobacteria in Oaks Bottom. One of the reasons that wasn't officially tested is, again, this is a recreational program. It's more of a bird watching, dog walking type area. And so signage was posted, but no samples were actually taken.
Good, thanks.
I can speak to that a little bit, a little more detail, Theo, but we had some reports of a bloom in Oaks Bottom earlier in the summer, and I think it was probably towards the end of August. I went out and collected some samples, and interestingly had quite a bit of Delicospermum. It looked different than Ross Island Lagoon, and obviously different habitat and everything there. But that was interesting. So we had a planktonic hab happening in the Oaks Bottom, but also notably, there was a lot of this benthic cyanobacteria formidium in the channel that connects the Oaks Bottom to the Holgate Channel, and it was alarmingly thick. And I think that's something that we've seen again and again in different restoration areas. I just can point to 3, at least right now off the top of my head, where we've seen quite a bit of soil movement and soil disruption from these restoration projects. And granted, it's probably going to take these areas a bit of time to heal over and recover from all the digging of the dirt and the disruption of those soils. But I think there's a lot of nutrients that leach out of those newly disrupted soils, and that's probably part of what's fueling the very thick growth of these potentially toxic benthic cyanobacteria. So it's we saw that that big red spot with this new USGS React tool that again was developed in Idaho, but at the same time you could see the big bloom up at Sturgeon Lake, up in Sovies Island and Vancouver Lake. So it's anybody's interested in harmful algae blooms. I know that most of the audience there are very interested in HABs, but this new tool is really has been a game changer. And let me know if you want to hear more about it. Thanks, Theo.
There's one last question.
Hey, Kurt. Hey, Nathan, this is Dan. I just wanted to ask a little bit about maybe local production of cyanobacteria, say like in Willamette Cove. There's a lot of focus, obviously, the Ross Island as a source, but I know we've talked about this in the past, but there's the potential for local productions, specifically because of the hydrodynamics, say at Willamette Cove.
Mm-hmm. Yeah, so with that React tool, you can zoom in and you can see areas of productivity. And actually, upstream of Willamette Cove, there are a number of docks and places where the water exchange might not be very good, and where a cyno population that's coming downriver from Ross Island, from River Forest Lake, from Clackamat Cove, upstream on the Willamette. There's lots of different places that could seed that bloom. And but we did see some areas of some returns, if you will, that suggested some chlorophyll levels in some of these areas by like marinas and docks and that sort of thing. So we probably good to be paying attention to that. And I think that's, when DEQ does their monitoring and their sampling, they're looking for places like that. Look, in the last, 5-6 years, probably there's just been more and more attention and interest in Ross Island Lagoon. And many of you, Dan Sabota and Desiree and other people there have been involved with some recent meetings and trying to figure out if there can be some restoration that's done to help with the blooms. But, certainly there's lots of, think about life history, and these cyanobacteria blooms, they often produce either spores or resting colonies that can reseed an area. But we do need to pay attention to these inoculation sources, because fixing the lagoon might not necessarily fix all the problems in the Willamette.
All right.
Yeah.
Thank you very much.
Willamette Cove last season was kind of an anomaly where we didn't see a bloom from Ross Island seeding the rest of the river. Instead, we saw it originate from Willamette Cove. So we're thinking it's potentially a source as well as Ross Island. Again, like Kurt said, if you kind of zoom in, you can get a pretty good picture with that 10-meter resolution of the coastline from Lumber Beach through Swan Island, Willamette Cove, all along Cathedral Park. Anywhere where there's like the shoreline, you're getting a lot of activity almost the whole year. And part of that might be anytime you have that water shore interface, the satellite's going to be a little less accurate, but there's activity going on all through there pretty much the whole season. Thank you.
All right. Thank you very much to all the speakers for this session. And we have now a break, and Theo